Hello everyone. Today we are going to learn about enzymes, general features, simple and conjugate enzymes in this PPT. Enzymes are mainly biocatalysts. They increase the rate of reaction by decreasing the activation energy. Majority of the enzymes are proteins, but sometimes RNA also act as enzyme like ribozyme. Enzymes are highly specific, that is specific reactions are being catalyzed by specific enzymes. Now in this graph, you can see how is enzyme decreasing the activation energy. This is a graph versus free energy versus time, that is progress of reaction. The red one is showing the uncatalyzed reaction where enzyme is not present. You can see the activation energy is high. So the reaction is slow. Now, here what you see in the blue one, this is a catalyzed reaction where enzyme is present. Here you can see the activation energy in the blue one has been decreased. How is this activation energy being decreased? Because enzyme is stabilizing the transition state. This is a transition state. Here it requires lot of energy. The reactants require lot of energy to reach the transition state. Now here the transition state is being stabilized by the presence of enzyme. Why? Because the enzyme is bringing the reactants close together in close proximity with each other and providing an optimal microenvironment. In this way, the enzyme is decreasing the activation energy and increasing the rate of reaction. That is, the progress of reaction is being fast. Enzyme has enormous catalytic power. It increases the rate of reaction by decreasing the activation energy, which we saw from the last graph. It changes the rate of reaction at which equilibrium is achieved. That is, it doesn't affect the position of equilibrium. It is only increasing the rate at which equilibrium can be achieved faster. Now here we see we can classify enzyme into simple enzyme and conjugate enzyme. Simple enzyme is only made of proteins like trypsin, pepsin. These are the enzymes which are only made of proteins or amino acid. Now conjugate enzyme is made up of proteinaceous part and a non-proteinaceous part. The proteinaceous part is called as apoenzyme and it requires some non-proteinaceous part. The non-proteinaceous part can be a coenzyme like NADH, FADH2, which are organic molecule and which are loosely attached. Or the non-proteinaceous part can also be a cofactor like metal ions Fe2 plus, Mg2 plus, Zn2 plus, Cu2 plus. Okay. Now, if the cofactor is tightly bound, then it is also called as prosthetic group. This figure just shows you two parts of your conjugate enzyme. The apoenzyme, the proteinaceous part, the cofactor, the non-proteinaceous part. Now apoenzyme and cofactor together are forming an holoenzyme, which is an active enzyme to which your substrate is bounding and your reaction is proceeding. The cofactors can be of two types like metal ions or it can be a coenzyme. Metal ions are like Cu2 plus, Fe2 plus, K plus, Mg2 plus. These are inorganic in nature and they take part in catalysis during the reaction. Now orgoenzymes are nothing but an organic molecules like NADH2, FADH2. These organic molecules are loosely bound to the enzyme. Usually vitamins are acting as a precursors for these coenzymes. Okay, like thymine, which is B1 vitamin, is acting as a precursor for thymine pyrophosphate, which is a coenzyme for pyruvate dehydrogenase. This chart is just showing vitamins which are precursor for the synthesis of specific coenzyme like thymine pyrophosphatase phase which is being synthesized by vitamin B1, that is thymine. Okay, riboflavin B2 which leads to the synthesis of FNN and FA2 which are being used as coenzymes for different reactions. Let's summarize. Enzyme increases the rate of reaction by lowering activation energy. Coenzymes are called dash molecule. Coenzymes are dash molecule. They are organic molecules. Removal of cofactor from conjugate enzyme produces apoenzyme, the proteinaceous part. The cofactor which are tightly associated with protein covalently or non-covalently are called prosthetic group. Prosthetic groups are tightly associated. Complete biological active conjugate enzyme is called holoenzyme.